Today we're looking at pathogens and disease for the GCSE topic. And so pathogens are microorganisms that cause disease, and they can affect plants or animals, that should say plants. They can be spread by direct contact, so touching somebody else, water such as cholera, contaminated water, or air, so things like coughs and sneezes, or they could be transferred by vectors. So vectors are an organism that can carry or pass on the disease to something else. So the spread of infectious disease can be reduced by simple hygiene measures like washing your hands properly, throwing away tissues, sneezing into your hand or a handkerchief, destroying vectors, so you could destroy the mosquitoes. You could isolate the infected individuals so they can't pass the infection, or you could give them a vaccination. So viral pathogens, viruses, the way they make us feel ill is they reproduce inside our body cells and burst them open, causing damage to the cells. So measles is an example of a disease caused by a virus, and the symptoms are fever, red skin rash, high temperature, fatigue, etc. So measles virus is spread by breathing in droplets from coughs and sneezes. Most people do recover well from measles, but it can be fatal if there are complications. So actually most children these days are vaccinated against measles. Another type of viral pathogen is HIV, which stands for the human immunodeficiency virus, which can then lead on to AIDS, which is acquired immunodeficiency syndrome. It can be spread by sexual contact or exchange of body fluids, so it could be when you share needles, etc. So at first HIV, when somebody initially gets infected by it, they will get flu-like illness, and then if untreated, the virus can actually enter the lymph nodes and attack the body's immune cells. So if you take antiviral drugs, that can actually delay this happening, so it can delay the onset of AIDS. During the late stage of HIV or AIDS, then the body's immune system is damaged and it can't fight off simple infections like coughs and colds, or the onset of cancer. Just to note that viruses can also infect plants, so an example of this would be tobacco mosaic virus. So now looking at bacterial diseases. So salmonella is an example of a bacterial disease which causes food poisoning, and it's usually caused by undercooked poultry. And the way that bacteria make us feel ill is they release toxins, which are basically like poisons, which get into our bloodstream, and they can also damage our tissues as well. So salmonella can be prevented by thoroughly cooking your food, wiping down surfaces with disinfectant after you've used raw poultry. And the reason it causes the cramps and the vomiting and the diarrhoea is because of the toxins. So next one, another bacterial disease, which is gonorrhea, which is a sexually transmitted disease caused by a bacteria, and obviously it's spread by sexual contact. The symptoms are thick yellow or green discharge from the vagina or penis, or pain when urinating. And it's usually treated with penicillin, but obviously a lot of bacteria are becoming resistant to penicillin now. So the prevention of gonorrhea would be a barrier method of contraception like a condom. Then you've got protists. So the main example you need to know about protists is malaria, which is caused by a protist, and the vector of malaria, the thing that passes it on from individual to individual, is mosquitoes. So mosquitoes are the vector. So a person would be bitten by the mosquito, and then it's obviously put straight into the bloodstream then. So malaria can cause severe fever, and if this reoccurs, it can be fatal. So the main way of preventing it is you can use mosquito nets to stop them actually accessing the skin. You could use insect repellents, or you can take anti-malarial tablets. Just to note, protists are single-celled like bacteria, but they are eukaryotes, so they do contain a nucleus. Okay, fungal diseases now. You've got rose black spot, which is a fungal disease of rose leaves, and it's spread by spores, which are carried in the wind, the rain, water, etc. And what happens is purple or black spots develop on the leaves, which turn yellow and drop early. And obviously, because they're turning yellow, there'll be less chlorophyll, which means there can be less photosynthesis, therefore less glucose will be produced, so less proteins can be made. So you usually find that they have stunted growth because they're reduced photosynthesis. And you can treat it using fungicides or by destroying or burning the affected leaves. So defences now against pathogens. You've got non-specific defences, which basically just try and stop pathogens from entering the body in the first place. So on the trachea, you'll have ciliated epithelial cells, which are little hair-like, the cilia are little hair-like structures which get have mucus on them, which trap any bacteria that you breathe in, gets trapped in the mucus, and then your cilia waft the mucus up and out of the airways, and then you can either cough that up or you could swallow the saliva, which would then go into the stomach, which isn't pictured here. So then go into the stomach, and the hydrochloric acid in the stomach will actually kill the bacteria. I've just noticed that's pointed to the liver. That's a really bad diagram. Okay. You also, on your skin is a barrier itself to any pathogens entering, but if you cut yourself, then your platelets in the blood would cause a blood clot or a scab to prevent any bacteria entering. You've also got enzymes called lysozyme in the tears, which destroy the bacterial cell walls. Your tears are actually also salty, which is a natural antiseptic because it dries out the bacteria. Got hairs in the nose, which stop pathogens entering. And your sweat also contains lysozyme, which is an enzyme which breaks down bacterial cell walls. So they're your non-specific defences. If the pathogen managed to get through all those defences and got into the bloodstream, then your white blood cells would help to defend against pathogens. And white blood cells can either do one of three things. It can either do phagocytosis, where they engulf the pathogen, they can release antibodies, or they can release antitoxins. So with phagocytosis, what happens is the white blood cell would detect, or the phagocyte would detect the pathogen, and then it would surround the pathogen, engulf it, and then these little vesicles called lysosomes would release the digestive enzymes and digest the enzyme into its amino acids, glucose, fatty acids and glycerol, and then the white blood cell can use those materials. Eventually, when it's digested quite a few pathogens, the white blood cell will eventually die because it will become full of toxins. So then the white blood cells can release antibodies, which are Y-shaped proteins which can attach to antigens. So antigens are the little marker molecules which are found on the microorganism, and they have the antigens are specific to each type of pathogen, a specific shape, and your white blood cell will release complementary antibodies that will attach to those antigens, and that can cause the pathogens to clump together, which then makes it easier for the phagocytes to come along and engulf them. And finally, you've got antitoxins, which are chemicals that can neutralise the poisonous effects of the toxins. If you wanted to boost your immunity, because the first time that a pathogen enters your body, it takes quite a long time for your white blood cells to to recognise that pathogen and then to produce the right antibodies. So it's quite a slow response, and during that time you'd probably get ill. once you've got the first infection, your body would produce memory cells which would remain in your body, which means that if you got reinfected by the same pathogen, you would then be immune to it, because your white blood cells would use the memory cells, would recognise the antigen straight away, and would produce a lot of antibodies really quickly to fight the infection. So actually the second time you get the infection, you won't get ill. And that is what is known as immunity. Now sometimes you wouldn't want to risk getting the primary infection and getting the symptoms if it was a deadly disease. So in that case you can use a vaccination. So a vaccination contains small quantities of dead or inactive forms of the pathogen, which can be injected into the body. And that helps to develop a natural immunity, so your body will produce the correct antibodies and produce memory cells, so that if you got the real infection, you would then be immune to it. So a weakened dead strain of the microorganism is injected. The antigens will still be there, so they will still trigger the production of antibodies by your white blood cells. They produce memory cells and remain in your bloodstream for a long time. So if you actually got the infection, you wouldn't get ill. If a large proportion of the population get vaccinated, it actually protects those that aren't vaccinated, because they can't spread the disease. And this is what is known as herd immunity, whereas most of them get vaccinated, then it actually protects the rest of the population. So treating diseases, you've got antibiotics such as penicillin, which antibiotics only kill bacteria because they work by destroying the cell wall. They don't work against viruses because viruses don't have a cell wall, and also it's very difficult to kill a virus because they live inside our body, so we'd have to kill our own cells as well. Antibiotics are very useful for destroying bacteria. However, there are resistant strains which are evolving. So MRSA is an example of a resistant strain of bacteria, which stands for methicillin-resistant Staphylococcus aureus, and it's quite prevalent in hospitals when people come out from operations and they've got quite a weak immune system anyway. The MRSA bacteria can then take a hold, and unfortunately there's no antibiotics that will work against it because it's actually resistant to most antibiotics. So, to reduce the rate at which resistant strains of bacteria develop, what doctors have started to do is not prescribing antibiotics unless they're absolutely necessary. And so they don't do it for non-serious infections, and they also make sure that they do not prescribe them for viral infections because they won't work. If they do give antibiotics out, it's very important that the patient actually completes the course of the antibiotics, because if not, what you end up doing is killing the weakest ones and leaving the most resistant strains behind, which is actually even worse. There is a required practical you do on agar plates, so you'll have a strain of bacteria, which is the brown bits, and then you'll put different types of antibiotic discs on which are soaked in antibiotic, and then what you'll find is a good antibiotic will actually kill most of the brown bacteria. So the bigger the area of the clear area means it's more effective. So probably this one and that one are really effective. That one's not very effective at all. That one's not done anything. That one's not really that effective. So the bigger the clear area, the better the antibiotic. So the independent variable, the thing you change, is the type of antibiotic disc. Dependent variable, the thing you measure, is the diameter of the clear area around the disc. And control variables would be things like same strain of bacteria, same volume of agar, incubate at the same temperature. Now you should always incubate at school at 25 degrees C, because if you do it higher than that, you risk the development of pathogens. So in industry they would probably use 37 degrees because it's faster. However, it's safer in schools to use 25 degrees C. So developing new drugs. So painkillers literally just treat the symptoms of a disease, but they don't actually affect the pathogen. They don't kill the pathogen, they just treat the symptoms. New antiviral drugs have been developed which can kill viruses without damaging the body's tissues, which is very difficult to find drugs that can do that. And new antibiotics needed to be produced because resistant strains of bacteria are developing. So traditionally, drugs were extracted from mainly plants or microorganisms. So digitalis is a heart drug that actually originated from foxgloves. Aspirin is a painkiller that originated from the willow tree. Penicillin was discovered by Alexander Fleming from the penicillium mold. So most of the time now, new drugs are synthesised by chemists in the pharmaceutical industry, but a lot of the time the starting point will still be a chemical that's extracted from a plant. So new drugs have to be tested and trialled to make sure they're safe, and so they need to be tested for efficacy, how effective they are, toxicity to check they're not toxic, and also to find the optimum dosage. So they're tested to see if it works and to find the optimum dose and to check that they're not toxic and there's no side effects. So once you've got a drug, you test it on a drug that you think might work. You would test it on cells and tissues. Then you would test it on animals, which are called preclinical trials. If it all works well on animals like monkeys and mice, etc., you would then start doing trials on usually healthy volunteers first, because they're more likely to be able to cope with the side effects. And you would usually do a double-blind trial. So a double-blind trial is where, well, first of all, I'll just tell you what a placebo means. So a placebo is a pill that does not contain the active ingredient or the active chemical. So it's basically like a fake drug, but it doesn't contain the active chemical, but it does look the same as the real drug. So a double-blind trial is where the patient doesn't know and the doctor doesn't know whether the patient has got the placebo or the real drug. So this reduces the psychological effects of you thinking that you're going to get better because you're taking the drug. And then you would allocate them randomly into two groups, either placebo or the real drug. It'd be double blind, so the patient doesn't know and the doctor doesn't know who's got the placebo and who's got the real drug. And if all that went well, you then test it on people who actually have the illness and check there's no side effects. So new painkillers are being developed to treat symptoms, but again, they don't kill pathogens. And new antiviral drugs are needed that kill viruses, but it's very difficult to kill a virus without damaging the body's tissues. Then finally, just a quick look at plant diseases. So the signs that a plant is diseased would be stunted growth, spots on the leaves, areas of rot or decay, growths, malformed stems or leaves, discoloration, and maybe the presence of pests. So to identify a disease, you can take a number of steps. So you could look at a garden manual or website. You could take the infected plant to a lab to identify the pathogen, or you could use testing kits which contain monoclonal antibodies. So, some examples of plant diseases. You've got tobacco mosaic virus, which is a widespread plant pathogen. It infects tobacco leaves in a mosaic pattern, funnily enough, and also can affect tomatoes. So it has a distinctive mosaic pattern of discoloration, and where there's less green colour and more yellow colour, that means there's less chlorophyll, which means less photosynthesis can take place. So you tend to find that they've got stunted growth. We've also got rose black spot, which is a fungal disease of rose plants. And aphids are the vectors. So aphids are small insects like greenfly, and what they'll do is they'll feed off the phloem of one plant, taking the sugars, and then they'll go to another plant and take the sugar from the phloem of the next plant and then directly infect the next plant. As well as communicable diseases of plants, you've also got non-communicable diseases of plants, which cause a range of deficiency diseases caused by a lack of mineral ions. So if there's a lack of nitrates in a plant, because nitrates are needed to make amino acids and therefore to make protein, you tend to find they have stunted growth. You've also got chlorosis, which is caused by magnesium deficiency, because magnesium is needed to make chlorophyll, so you tend to find they have less green colour and more of a yellow colour. Got a range of plant defences, so physical defences like cellulose, cell walls to stop herbivores from eating them, a tough waxy cuticle that's difficult to chew through. There's also layers of dead cells around the stems, so the bark on a tree, when that falls off, that'll take any pathogens with them. There's also chemical defences, such as having like a sour taste or a not nice taste, or some actually have antibacterial chemicals such as tobacco or foxgloves, which will kill any bacteria on the plant. And then finally, there's other mechanical adaptations like thorns or hairs, the herbivores from eating them, or from laying eggs on their leaves. And then you've also got some kind of mimicry. So some white dead nettle looks very similar to normal nettles, but they actually don't sting. But it does deter the herbivores from eating them, or the insects from eating them, because they think it is a nettle, so they won't bother eating it in case it does have a sting, but it actually doesn't have a sting.